Arkat Abzhanov and Cliff Tabin have spent years trying to find out exactly how those Galapagos finches got their different beaks. Their starting point was what they had learned from Darwin himself. Their beaks were vital to the bird's survival. On an island where the main food was seeds, finches had short, tough beaks for cracking them open. On an island where the main food was from flowers, birds had long, pointy beaks for sucking up nectar and pollen. And they knew something else. The finches are born with their beaks fully formed. So the answer to why they had such different beaks must lie in something that happened to them as embryos in the egg.
Something amazing is happening inside those eggs. Genes are turning on and turning off. And depending on exactly how they turn on and off will determine what type of finch is formed.
To find out just what was going on, the researchers first had to collect some eggs. Another nest already has 3 eggs. He takes one for his research, as he knows the mother will lay a replacement. The team collects several eggs, with embryos at different stages of development. That way they will be able to chart exactly how the different beaks grow. Back in the lab, they can begin the process. This cactus finch embryo is well on the way to its signature long, pointy beak. And this ground finch embryo is growing a short, thick beak.
What we wanted to do was try and understand the genes that were involved in making the beak the way it was, making a big, broad, thick beak different from a long, thin beak or a short, thin beak.
They concentrated on a group of genes known to control the growth of birds' faces. As they looked, they saw something intriguing. One particular body plan gene became active in the ground finch with the short, thick beak on the 5th day of development. But it didn't go to work in the cactus finch with its long, slender beak for another 24 hours. This was a revelation. The same genes were responsible for the beaks in all types of finch. Any differences were in timing and intensity.
We've got it. We nailed it. It's the same genes in making a sharp, pointy beak or a big, broad, nut-cracking beak. What's essential, what makes the difference, and all the difference, is how much you turn a gene on, when you turn it on, when you turn it off.
And the revelations didn't end there. There was something special about this gene. Like all body plan genes, it doesn't actually make the stuff of our bodies. It didn't make the cartilage for the finch's beaks. It throws switches. And the switches then turn on or off the genes that do make the beak.
These are a different type of gene. They're genes that boss other genes around.
Scientists now realize that not all genes are created equal. Some make the stuff of our bodies, and switches are needed to turn many of these stuff genes on and off. The body plan genes are what throw these switches, which tell the stuff genes what to do and when. This subtle choreography can have profound effects on how different animal bodies are formed. And this knowledge is helping us solve perhaps the biggest Darwinian puzzle of all, the mystery of the great transformations. Darwin had seen equally persuasive evidence in embryos. Those slits in the ear of all land creatures, even humans. In us, they become tiny bones in the inner ear. But in fish, they become gills. A tantalizing hint that land animals must be descended from fish. But the stumbling block has always been how. How could a fish develop legs and walk on land? Darwin had no idea. But Neil Shubin was determined to tackle that problem.
It captured my imagination. I mean, here's a fin, and on the other side was a limb, and they looked different in many ways. And I thought, well, what a first-class scientific problem to devote my research to. And I've been devoting pretty much my research to it ever since, over 20 years.
The first stage in Shubin's quest was to find a fossil. If Darwin were right, somewhere out there had to be a transitional form, a fossil that was part fish, but had the beginning of legs. But where to look? He had one clue. The fossil record shows that creatures with legs first appeared some 365 million years ago. Before that, they were only fish. So summer after summer, Shubin set up camp on Ellesmere Island, just a few hundred miles from the North Pole. After 3 days, they still hadn't found anything. Then, just when no one was expecting anything to happen,
A colleague was cracking rocks, and I was working about 5 feet from him. And I hear, hey guys, what's this?
Well, look what we found. Yes, it is.
Sticking out of the cliff was the snout of a fish. And not just any fish, a fish with a flat head. And by seeing a flat-headed fish in rocks about 375 million years old, we knew we had found what we were looking for.
a flat snout with upward staring eyes, the signature of an animal that pushes its head out of the water. And for that, it would have needed something like arms. Back at home, Shubin and his team got to work, examining their 375 million-year-old fossil. They named their new finding Tiktaalik, an Inuit word for a freshwater fish. Tiktaalik is a perfect transitional form. Much of its body is that of a fish. It's covered in scales. But it also had something very unfish-like. An arm-like fin, or perhaps a fin-like arm. Tiktaalik had the bone structure that is seen in the arms and legs of every four-limbed animal. One big bone at the top, two bones underneath, leading to a cluster of bones in the wrist and ankle. It's the same pattern that is found in everything from sheep to sheepdogs, to Shubin himself.
You now have an animal that can push itself up off the substrate, either on the water bottom or on land.
One obvious question was, why had Tiktaalik evolved this new structure? One possible answer is suggested by other fossils found near it.
There are large predatory fish about 10 to 15 feet long living alongside Tiktaalik.
Tiktaalik was prey. To survive, it had few choices.
You can get big, you can get armor, or you can get out of the way.
Shubin thinks Tiktaalik got out of the way. With those arm-like fins, it could have dragged itself to safety on land or in the shallows. But this was only half the answer.
What it doesn't show us is the actual genetic mechanism, the genetic recipe that builds a fin into that which builds a limb.
At 375 million years old, Tiktaalik's DNA had vanished long ago. Shubin needed a next of kin, a fish relative that was still alive.
What we needed was a creature that was in the right part of the evolutionary tree, but also a fish that has a very fleshy fin. So the search was on.
The paddlefish.
Paddlefish is a really weird fish. They develop this really long snout.
Living in the shallow waters of the Mississippi, it's also a living fossil. Scientists have spent years working out the relationships between different species of fish, and they know that the paddlefish is one of the last survivors of the class to which Tiktaalik once belonged. But unlike Tiktaalik, the paddlefish is in plentiful supply. Intriguingly, even though Tiktaalik is extinct, the paddlefish is actually the more primitive form. Its fins bear far less relation to an arm or leg than Tiktaalik's. And because they are related, the two kinds of fish should share the same genes. So Shubin began looking at paddlefish embryos, hunting for the genes that built its fins. And soon, he zeroed in on one particular group of body plan genes, called Hox genes. Hox genes have been found in all complex animals, from the velvet worm that dates back some 600 million years to the modern human. And in all that time, the letters of their DNA have remained virtually unchanged. They are aristocrats of the gene community, near the very top of the chain of command. They give orders that cascade through a developing embryo, activating entire networks of switches and genes that make the parts of the body. They are absolutely critical to the shape and form of a developing creature.
These genes determine where the front and the back of the animal is going to be, the top, the bottom, the left, the right, the inside, the outside, where the eyes are going to be, where the legs are going to be, where the gut's going to be, how many fingers they're going to have.
Shubin found that Hox genes had a key role in the formation of paddlefish fins. One set of Hox genes orders the first stage of fin development, a sturdy piece of cartilage that grows out from the torso. Amazingly, in all four-limbed animals, even us, exactly the same genes create the long upper arm bone. In the paddlefish, another set of Hox genes command the next stage of fin development. Again, exactly the same genes control the growth of our two forearm bones. Finally, the same genes, working in a different order, make the array of bones at the end of the fin. The same sequence of the same genes makes our fingers and toes. This was a massive revelation. Suddenly, the origin of creatures with arms and legs didn't seem such a huge leap after all. If the same genes were at work in Tiktaalik, then many of the genes needed to make legs and arms were already being carried around by prehistoric fish. All it needed was a few mutations, a few changes to the timing and order of what was turned off and on, and a fin could become a limb.